Welcome to our Minds, Technology, and Society seminar on April the 9th, 2018. We thank the Gleschko and Samuelson Foundations for funding and the School of Social Sciences, Humanities, and Arts. Our speaker today is Dr. Kelly Weinersmith, who is currently an adjunct assistant professor at Rice University in BioSciences and off on some sort of adventure with the Grand Science Farm, which you may or may not talk about. Kelly and I went to grad school together at UC Davis. She did her PhD in ecology. She is also recently the author of the New York Times bestselling book, Soonish, 10 Emerging Technologies That Will Improve and or Ruin Everything.
Nice. Wow.
Really glad I remember that. Kelly studies parasites and how they're awesome. So I'm going to stop talking and leave it to Kelly.
All right. Thanks. I'm excited to be here. I got to talk to some of you today and I had a blast, so I really appreciate everybody's time. So today I'm going to tell you about a topic I love, which is parasites. So parasites are everywhere, but you don't see them because they're sort of out of sight and out of mind. So for example, in Southern California estuaries, the biomass of parasites in those systems can exceed the biomass of top predators. For example, the biomass of trematode parasites, in some cases in some of these systems, exceeds the biomass of the fish and the birds out there. So you don't see them, you maybe don't think there's that many, but there's a lot of them out there by biomass. And those trematodes in particular can manipulate the phenotypes of some of their hosts. So for example, they control the behavior of they castrate a lot of the snails out there. So those snails are never going to produce their own offspring. They are essentially the extended phenotype of the trematode parasites moving around in these salt marshes. So parasites make up a lot of the biomass and they control a lot of the free living biomass also. So you can imagine this could have important implications for food web dynamics, the evolution of host behavior, and I'm particularly interested in these parasites that manipulate host behavior, and I'm also interested in what these parasites can teach us about links between the brain, the immune system, and behavior. So this is my new favorite quote. Tell me what parasites you have, and I'll tell you who you are. Dr. Claude Coombs said this. It's a play on a foodie quote, so it's a little less original than I originally thought. But anyway, so the point of this quote is twofold. The behaviors that a host engages in determines the kinds of parasites they encounter and how often they encounter those parasites. On the flip side, after hosts get infected, sometimes their behaviors change. So there's this intimate link between behavior and parasitism in a lot of different systems. So actually I'm going to step back real quick and say that I would love for you to interrupt me at any point throughout the talk to just ask any question that you have, because I'd love to chat with all of you since you are not my usual audience. I'm excited about the questions you're going to ask and the perspectives that you can give me. Okay, so for example, behavior can influence parasitism. These are guppies. Guppies are infected by lots of parasites. One of them is an ectoparasite, a parasite that lives on their skin, called Gyrodactylus turnbully. This parasite reproduces on their skin and gets transmitted from one fish to another when the fish rub up against each other. So the more in this system, the more social fish are more likely to have this parasite. And in this system, the females are more social. So in this study in particular, the females were 4 times more likely to have acquired this parasite than the males were because of the behaviors that they engaged in. So there's an example of behavior influencing parasitism. On the flip side, parasitism can influence behavior. It can do this in a number of ways. One of those ways is pathology. So the host gets sick, that's bad. The host starts changing their behavior in ways that are bad for both the host and the parasite. That's what pathology is. Additionally, the host can change their behavior in ways that are good for the host, but bad for the parasite. For example, they can induce a behavioral fever. They can go to an area that's a little bit warmer than usual in the hope of killing the parasite, or in the hope of just reducing the cost of infection. Third, parasites can manipulate host behavior. This involves changing behavior of the host in a way that is good for the parasite, but bad for the host. One of the most popular examples of this are the zombie ants. So this is an ant in the genus Compotus. This particular story happens in the rainforest. These ants usually live in colonies up in trees, but they have to go down on the forest floor to forage. So they walk along the forest floor. They can accidentally get infected by a spore of a fungus called Ophiocordyceps unilateralis. The fungus gets inside the body of the ant behaves normally for a little while, and then when the fungus is ready to reproduce, It causes the ant to leave the colony, stumble down the tree as though it's drunk, walk along the forest floor until it finds a suitable sapling, and then on the northwest side of the plant, around solar noon, at about 25 centimeters up off the forest floor, it finds a leaf. It crawls along the bottom of the leaf. Its mandibles bite down in the leaf vein, and then the ant can no longer use its mandibles. It stays there till it dies. And the fungus creates a stalk, and from the stalk rain the spores of the parasite down on the forest floor below, so the next group of ants can come by and get infected. So maybe you noticed how many qualifiers I had to add for this manipulation. The northwest side of the plant at solar noon, about 25 centimeters up. This is amazingly specific in ways that like totally blow my mind. And it turns out a lot of that specificity is really important for the fitness of the fungus. So if you take a leaf, with an ant and a fungus on it, and you move that leaf to the forest floor, the ant disappears. Presumably because an invertebrate predator like a spider came by and ate the ant, and that kills both the ant and the fungus. So that's bad for the fungus, so you've got to be higher than the forest floor. But if you move too far above 25 centimeters, temperature and humidity are no longer good for fungal growth, and this stalk can't form and the fungus can't reproduce. So this 25 centimeter zone is really important for the fungus. So if you were to take one of these ants and bring it to the lab of a neuroscientist, you'd say, hey, ants have pretty simple brains, I think, relative to our own brains. Do we know how to recreate these behaviors in an ant that the fungus is able to produce? And the answer, as far as I can tell, is no. Even with a sort of simple organism like an ant, we don't know as much as a fungus. And of course, that's like way anthropomorphic. I don't think the fungus knows what it's doing. But before we feel too bad about not being as smart as a fungus, we should keep in mind that this fungus has probably had about 48 million years to benefit from the process of natural selection to tinker with the host brain. So I mentioned that the ants use their mandibles to bite down around the leaf vein. They leave very distinctive marks called a death grip, because everything about this system is epic to the max. So they leave this death grip, and David Hughes and his collaborators found fossilized leaves that are 48 million years old, so from the Eocene, back when a bunch of mammalian orders were sort of showing up. It looks like the fungus was already getting the ants to sort of bite down around major leaf veins. So a long time for these parasites to benefit from the process of natural selection. So I'm excited about these parasites as a way to understand links between the brain, immune system, and behavior, because apparently they've learned a lot that maybe we can learn from them. So my research that I'm going to talk about today, so first I'm going to talk about a new system that we discovered recently. So this is one thing, one of my goals in life is to discover new systems and actually quantify fitness benefits to the parasites arising from changes in host behavior. And one of the reasons I think that's important is because we've been fooled before. So it's really tempting when you see that a host acts weird when it's infected to call it manipulation and come up with a story for how it benefits the parasite. So for example, mosquitoes that are infected by malaria, malaria is transmitted when it bites people, and then when a mosquito bites an infected person, it sucks up the parasite goes through some stages, and when it's ready to reproduce again, it moves into the salivary glands. So when the mosquitoes get infected, there's that period where the parasite is in them but cannot be transmitted. And during that period, mosquitoes bite less. And you can say, well, that probably benefits the parasite because if mosquitoes are biting humans, they've got some chance of getting smacked. That's bad for the mosquitoes and for the parasite. And then when the mosquitoes get to the point where they can be transmitted, where they're in the salivary glands ready to go, the mosquitoes start biting more. So of course that sounds like something that's good for the parasites too. When the parasite is ready to bite, the mosquito does something stupid and starts biting more and more humans. And so for a long time we were like, oh, malaria manipulates. But it turns out, so Lauren Cater injected mosquitoes with heat-killed bacteria, and they do the same thing. So this is a behavior that's driven by the host, not by the parasite. It might still benefit the parasite, but my point is that there's a lot of things that we call manipulation, and I think we need to be a little more careful before we throw that word around. Question?
So why does it correlate so perfectly with the gestation of the parasite?
Yeah, great question. I don't know. I don't know why heat-killed bacteria do that, but they do. And so people are trying to figure out how it might benefit the host to behave that way. It's possible that like when you first get infected by something, maybe you spend a couple days in bed and then at some point you're like, I can't stay in bed forever, and then you gotta make up for lost time. Obviously that's like super anthropomorphic, but the point is you can come up with, You can come up with something to say, but I don't think we actually know the answer. Okay. So then after that, I'll talk about a new research, or a second research system that I work on. In this system, I like looking at how behavior influences encounter rates with phenotype changing parasites. I'm interested in what behaviors are changed and how extensive these behavioral changes are. I'm interested in looking for feedbacks between behavior and parasitism, because this can have important implications for the variability of host behavior in a population. And I also study the mechanisms through which parasites manipulate host behavior. Depending on how many questions I get today, I might not get to this part. So we'll see. We'll maybe get to point 5. Okay, so let's start by talking about this new study system. I like to start off by thanking the people who helped out on the project. So the folks up top helped with the logistical work. These were my funding sources. These were my amazing collaborators. The artwork is done by this amazing cartoonist named Boulet. He's a French cartoonist who I can't believe we got to help us with this, but we did, so that's great. So the story starts with this guy. This is Scott Egan. He's a professor at Rice University, and he loves gall wasps. It is so fun to talk to him about gall wasps. You can't not love gall wasps when you're done talking to him about them. And so gall wasps are these little insects who lay their eggs inside the developing parts of plants, and then they induce growth in the plants that benefits the parasite and is bad for the plant. So essentially, these wasps are manipulating the phenotype of the plant. That's not actually the manipulator that we're interested in this story, but this is the first level of manipulation. So for example, here are a bunch of growths that were induced by multiple eggs that were laid into the plant. And the reason that these growths are made, one of them, is because it seems to protect these wasps from parasitoids. So there are insects that would like to also lay their eggs into these compartments so that they can eat the wasps developing inside. But if there's this big growth surrounding the developing insect, then the ovipositor of the parasitoid has a big space that it has to search to get to it. And so that's one of the reasons these growths are induced. So he was walking around in Florida with his daughter on vacation, and they were passing Quercus geminata. These are sand live oaks. They're like a quintessential southern tree. I think they're absolutely beautiful. And he saw some galls, and he always has Ziploc bags in his back pocket for when this happens. And so he pulled one of his Ziploc bags out, took notes on where he was, stuck the galls in a bag, brought them back to the lab. And the galls that he collected were produced by the crypt gall wasp. And I can't tell you how excited I was when I found out that was the name of the organism, because I wanted to work in a system as epic as David Hughes' zombie ants. And I was well on my way. So this is what the host looks like. This is a drawing of the adult about to lay an egg inside a developing stem. And so the gall wasp undergoes development in this compartment called the crypt. So it induces the production of a compartment that is lined with nutritious tissue. And as it goes through development, it eats that nutritious tissue and it lives in this compartment. And so that's a cross-section of what the crypt looks like while the insect's developing. And these are 2 crypts. So in this particular branch, I took a razor blade and cut off the bark. So this is one adult wrapped up in the crypt developing, and this is another adult wrapped up in the crypt. And if everything goes well for them, then they become adults and they excavate an emergence hole out of the crypt and they leave. So there's no way to get out of the crypt unless the wasp creates an emergence hole. And this is, if you look at a stem, this is what it looks like when you have a crypt from which the adult has escaped. This all makes sense so far? Okay, so Scott brought a bunch of these home. He stuck them in a clear Solo cup on his desk and put a coffee filter on top of it, because that's the kind of equipment that you need when you work in this system, which is one of the reasons I love this system. And what he noticed was that there were a lot of these holes suggesting that the adult got out of the crypt successfully, but there were also a lot of these other holes where, do you see, in the middle of this hole, there's a little bit of brown? That's the head of the gall wasp, and then there's a little bit of gray up top, that's part of its eye. So these wasps made some emergence holes, but then got stuck like this. And that kept happening, and that's not something that usually happens. And so he wondered what was going on, and he actually checked these cups right after I did my job talk at Rice for the Huxley Fellow. And when I got to Rice for that position, he said, well, do you think that this is manipulation? And I said, well, yeah, maybe, I don't know, let's check it out. And so if you dissect open these holes that are plugged by the heads of what I'm now just going to call the host to keep things simple, inside of the crypt is a developing parasitoid And surrounding the parasitoid, all of these little shiny bits are bits of the exoskeleton of the host that have now been strewn around the crypt after the host finished eating its insides. That's what parasites do to get nutrition. And so there was this very strong correlation between the host getting stuck in the crypt and the presence of a parasitoid living in the crypt. And if you let those cups sit long enough, this is what comes out. So this is what that parasitoid looks like when it becomes an adult. And it's beautiful. Like, it's iridescent, it looks like it's wearing socks. I think it's super cute. And if you key it keys out to the genus Euderus, but it didn't key out to any particular species, so it hadn't been described by science. So we got to name this critter. We called it Euderus Set, after the Egyptian god Set, who's the god of like chaos and stuff, and he trapped his brother Osiris in the crypt. It's very metal. And then eventually he ripped Osiris to pieces and threw his body parts, and that's kind of what looks like happens inside of the crypt, because this guy's a messy eater and the bits of the host are sort of all over the crypt. And so anyway, we were pretty proud of ourselves, I'm gonna be honest. And so this is another photo of what it looks like from a different angle. They're like amazingly iridescent. The direction of the light changes the color. That's the definition of iridescence. They're absolutely gorgeous. And so what happens in this life cycle is that the wasp lays an egg inside of the crypt where the host is developing. We don't know exactly what stage the host is when this happens. We're trying to get the timing down in nature, but at some point they lay an egg in there. And so you've got a parasitoid developing inside, and at some point they induce the host to create an emergence hole, but instead of emerging, they get stuck, and they plug the hole with their head. And at that point they die and the host eats their insides. And when the host is finishing developing, it pops out through the head of the host and goes off about and does its business. So here is the head of an insect. Here's an eye, here's an antenna, here's a big gaping hole where the parasitoid came out. Again, very metal. So we love this system. So one of the first things we did was just sort of try to describe what we were seeing. So we went out into the field, and on 2 different occasions we collected stems that clearly had a lot of these hosts plugging the holes with them. And we numbered every single hole so we could track it over time. And we measured the diameter of these holes. And so one of the first things that we noticed was that the diameter of the emergence holes are smaller when the hole is made by an infected host than when it's made by an uninfected host. So you can just kind of see that here, like the diameter there is bigger than the diameter there. And it didn't matter which of the sessions during which we collected the stems we looked at. It's always the case that the uninfected ones are making bigger emergence holes. So then we started asking, well, how does this benefit the parasitoid? What is the benefit here? So we had a couple different predictions that we wanted to test. So first we thought, okay, well, why is it important that the host's head plugs the hole? So we thought that might be important because if the host created the emergence hole and then crawled back into the crypt, then that would allow air into the crypt. And these developing stages of insects are pretty sensitive to humidity and temperature. So we thought that maybe plugging the crypt with the head was particularly important for making sure that temperature and humidity stayed the same in the crypt. So to test that hypothesis, we had a treatment where we had heads plugging holes, we just left those alone. For another treatment, we took needles and we poked holes in the heads of the host to allow air inside of the crypt. And so we thought that the fitness of the parasitoids in that hole should be lower, but it could be lower either because air got in there and that's bad, or it could be lower because it's not good to poke a needle around the hole when things are developing. And so we had another treatment where we used the pin to poke a lot of holes, and then we covered the hole with a tiny piece of bark, and that would keep the internal conditions in the crypt constant. And if we ended up with a lot of death in there, it would suggest that our needle was the thing that was killing everyone, killing the parasitoids. And then our second hypothesis was that what's important here is the host making an emergence hole, because the parasitoid is not good at doing that itself. So to test that hypothesis, we took a little bit of bark off of some trees, and we covered the holes with that bark. We put tape around the bark, but not over the hole. So if a parasitoid wanted to get out, it would have to get out through the parasitoid and then through a little bit of bark. Does that make sense? So this probably underestimates how hard it is for the parasitoid to get out, because they have to get out through woody tissue and bark if there's no help from the host. And here they just need to get through a little bit of bark. But it was raising the bar a little bit, so we decided that was a good place to start. So our prediction was that the control should have the highest survival. And then any of the two situations where we resealed the crypts with bark should have lower survival. But we were particularly interested in what was happening with adults for this prediction because the adults are the ones that need to get out. Okay, so what did we find? So this is the proportion of parasitoids that survived. The darker gray is the proportion that survived. The numbers are the numbers of parasitoids that were in each of our different treatments. And what we found, so let's look at across all life stages, when you breach the crypt, so when you poke it with a needle, that doesn't reduce survival that much relative to the control treatments. The 95% confidence intervals overlap with 0. Maybe it reduces fitness a little, but probably not a ton. What really seems to reduce fitness is any situation where the parasitoid needs to get through any bark. So the reseal treatments have negative log likelihoods for survival that don't overlap with 0. And that's mostly a story about what's happening with the adults. So if you look at the graph over here, we had pretty good fitness for the control individuals that survived to adulthood. Breaching reduced fitness a little bit, but not a ton. But anytime they needed to get through the bark, they had about a 3-fold decrease in survival. So needing to get through just a little bit of bark makes them 3 times more likely to die trapped inside of the crypt. One of the things that we weren't able to test in this experiment that we're interested in following up on is the benefit of having the crypt covered by the head in terms of avoiding hyperparasitoids. So it's a tough world out there. There are actually parasitoids that go after Eudera set. And if the crypt is covered with the head of a host, maybe it's harder for those hyperparasitoids to get in there. So that might be part of the benefit, but we haven't been able to test that in the lab yet. So it's still possible that having the head covering the crypt is important, but it doesn't seem to be important for maintaining internal temperature and humidity conditions. Okay, so that is not to say that the parasitoid can't ever get out of the bark on its own. So this is one of the treatments where the parasitoid, so this is the tape that was holding a little piece of bark over one of the holes where the host head was covering it. And that circle is where the parasitoid managed to get through the bark and get out. So they can do it sometimes on their own. They're just much less likely to be able to do it when they have to get through any bark. So they're pretty crummy excavators. So we wanted to know how common this was. And so one of the things we did was search various sites where you find Quercus geminata, the host tree that I was talking about. And Quercus virginiana is a very closely related oak tree. And so Basedia pallida, the host wasp, can live on both of those. And so we found this happening throughout the range. We found some evidence of it in a Smithsonian sample from 1983. And then we also found it on some closely related some other oak trees. For example, Quercus nigra, Quercus lobata, Quercus douglasii. They probably have different Basedia species, so different species of the gall wasp. And if you have a different species of a gall wasp, you probably have a different species of parasitoid also. So this thing that we found happening might be happening in a lot of backyards across the United States. And so we're pretty excited about trying to figure out how widespread this is and get to name more parasitoids, because that was fun. Interestingly, these samples that we looked at the American Museum of Natural History were collected by Alfred Kinsey. I don't know if anyone recognizes that name. So before he was like the guy who was famous for being like the guy who studies human sexual behavior, he was like a rabid fan of cyanipid gall wasps. And he collected millions of them and put them in the American Museum of Natural History, and a bunch of them have not been sorted through yet. So anyway, weird factoid that I thought was interesting and must share every chance I get. So in this system, we found this cool new example of manipulation that had apparently been sitting in museums for like 100 years and could have been discovered. Was like literally happening on the campus that I worked on, but no one had noticed it until that time, which I was excited about, because when I started working in this field, I was like, oh, have we discovered like all the cool examples of manipulation? But there's probably a lot more out there. That's exciting. And so it looks like the parasite gets about a 3-fold increase in survival arising from this behavior. Other things we're looking at is trying to figure out all of the timing of when this stuff happens. When does the parasitoid lay its egg in the crypt with the host? How does it manipulate host behavior? So we're working on getting the genomes and just trying to figure out what hormones that we should be honing in on. And additionally, we're interested in quantifying the whole parasitoid community that infects that cyanipid gall wasp, the crypt gall wasp. So these parasitoids also came out of crypts. None of these parasitoids were associated with the head plugging thing that we talked about, So they don't seem to be manipulating host behavior, but they are also laying eggs in the crypts with Basidia pallida. So it is probably pretty miserable to be Basidia pallida, but here's a lot more beautiful wasps that we get to work on, so that's pretty exciting. So anyway, that is the end of the first story. Oh yeah, see, it looks better over here. There's a lot more beautiful iridescence. We want to know why parasitoids are so iridescent, so we're working on trying to figure that out also. But anyway, okay, so that's the end of this first story. Does anybody want to interrupt with questions now about that first part? Just awestruck, Paul?
Yeah, so the manipulation here is that, because the host here always makes a hole.
Mm-hmm.
But so the manipulation here is basically to stop it from finishing its hole?
Yeah, so to figure out the timing for when it does it, to make the hole smaller than usual, and to get it to essentially not complete it and get stuck before it leaves.
Yeah, I mean, could the mechanism for that be as simple as just basically like lets it start making the hole and then kills it before it finishes, or is it?
Yeah, so it could be. So what if that were the mechanism, which it could be. I think that we would expect that sometimes they would blow it and would do it a little too late, and the host would get out. We never find the parasitoid in a crypt where the host got out, but I suppose in any mechanisms they could through any of the mechanisms they could mess up the timing. But so yes, that's something that we are planning on looking at. It could also be something like creating a small amount of whatever hormone the host produces when it's time for it to leave, like an insufficient amount for it to finish the job. So yeah, it could be any of those things.
Just a quick question about your term manipulation throughout these examples.
Yeah.
Because I was going back to the mosquito that you mentioned with malaria.
Mm-hmm.
It seems like you could induce a small sort of, it sounds like an immune response of sorts, right? So if it's responding to dead bacteria of some sort, and it's responding to malaria of some sort, both cases by not flying out and biting more things, that sounds like a manipulation to me, but you disqualified it as a manipulation. I was curious, what's the dividing line there?
Okay, so in this system, to 100% wrap up the case that it's manipulation, we need to find the mechanism, for sure. But so the criteria that we usually use to say like this is definitely manipulation is one, it has to be driven by the host and driven by the parasitoid or the parasite and not the host. It's hard to imagine any way that this benefits the host. And you have to be able to show that it actually increases the fitness of the parasite. So those are sort of the two things that we really look for manipulation systems. We've got one of them here, and now we're working on nailing down the other. Who knows how long that will take us. Okay. Yeah, sure. Any other questions? Okay. All right, so my next system is a trematode parasite that infects California killifish. I'll start by thanking the many collaborators that I had for this part of the project, including Andy C., my PhD advisor, and Ryan Heckinger, now at Scripps, who worked with me on just about all aspects of this project. And my research funding, or the funding sources for both the experiments and for myself. Okay, so let's talk about the life cycle of this parasite. So this is a trematode parasite. It, as an adult, lives in the guts of predatory birds. They find a mate. They produce eggs. Those eggs pass with the bird's feces into salt marshes in Southern California and Baja California. Snails accidentally consume those eggs. And the snails, as if it wasn't bad enough to accidentally be consuming poop and parasites, they are subsequently castrated. So they will never produce any of their own offspring. They will spend the rest of their lives, which can still be up to a decade, producing parasites. So their gonad tissue is now controlled by parasites. The parasites reproduce asexually. They produce a free-living stage, known as a cercaria, that leaves the snail, swims around, and when it encounters a killifish, we think it burrows through its skin, follows nerves up to the fish's brain, and then forms a cyst on top of the brain of the fish. Every cyst that you find on a fish's brain was independently encountered while they were swimming around, because the parasites don't reproduce at this stage. So if you find 1,000 parasites, each one were acquired individually, as opposed to one that reproduced 1,000 times. The killifish, so no reproduction for the parasites at that stage. When the killifish is consumed by a predatory bird, the parasite can complete its life cycle. But if the fish is not consumed by a predatory bird, the parasite will die. So the fish has to die in a very particular way in order for the parasite to complete its life cycle.
You said if you find 1,000 separate incidents, do you find it?
Oh, yeah, we'll get to that.
frequency? Okay.
Oh yeah, 1,000 is like low. It can get up to like 8,000. In fact, here's a photo of that. And so to orient you, these photos are a little bit, you can see it a little better here. So the like iridescent greeny kind of thing down here, that's one fish eye. Up top, that's the other fish eye. Down here is the brainstem. Here you've got the optic lobes. And maybe you can see over in this area, in the space between like the skull and the brain, there are all of these little white dots. Those white dots are the excretory bladders of the fish. And those excretory bladders are surrounded by like a sort of clear space. That's the rest of the parasite. So pretty much this entire brain is covered in parasites that are packed in next to one another, literally touching each other. Here they're like, you know, filling the space between the brain and the skull. There's none up there, but that's just because I'm clumsy and my equipment touched the brain and kind of knocked it off to the side. But this adult had, I think, 1,500. I've caught fish that still had their yolk sacs, which means they had hatched in the last 2 to 3 days. They were already infected. By the time they reach adults, the question isn't are they infected or not, it's do they have 1,000 parasites or 8,000? It will be somewhere in between there. It varies a little bit depending on the population.
Do the parasites never kill the fish?
They don't seem to. So every once in a while, we'll see a little bit of evidence of brain damage. So like right over there, you can see maybe there's a little bit of blood, like a little bit of a hematoma. But one of the amazing things about this system is that the fish mostly act normal. Like they forage normally, they're not swimming on their sides. Like you'd think that with this many parasites, they'd be like swimming on their sides, dying on the floor. But no, for the most part, they act normal. And so this is a population of infected killifish swimming around in Southern California estuaries. You know, they look normal, they're checking out the camera. But they also do these things called conspicuous behaviors. So there's going to be a fish over here that's going to flip on its side. You're going to see like a flash of light. There it goes, and there it goes. So the more of the parasites they have, the more often they do these conspicuous behaviors, and the more likely they are to get eaten by predatory birds. And this is not work that I did. This is work that was done before I came along. So this is pretty good evidence that manipulation is happening. We're working on nailing down the mechanism, but at least evidence that that more infected fish are more likely to get eaten by predatory birds, and the more parasites they have, the more often they do these conspicuous behaviors. So a woman named Jenny Shaw, for her postdoc, started trying to nail down the mechanisms, and she noticed that infected fish differed in serotonergic and dopaminergic activity in particular brain regions. And in teleost fish, we know that these neurotransmitters are associated with things like boldness, social behavior, exploratory behavior, activity. So I was like, oh, I bet all of these subtle traits are changing in response to infection, but nobody's noticed it because these fish are doing these like really obvious conspicuous behaviors. That's what we've been focusing on. Probably these more subtle like personality traits are changing also. So for my dissertation, I wanted to look at how parasitism changes behavior, focusing on these kinds of traits. I'll talk about one of the like 3 studies I did looking at this. This study was done with a woman named Violet Compton-Rennick. She was interested in how pesticides change the behavior of killifish, because this is also a common stressor in Southern California estuaries. So we looked at interactions between 3 different concentrations of a pesticide called chlorpyrifos. It's an organophosphate pesticide. And then we looked at how the pesticide changed behavior, how the parasite changed behavior, and how interactions between those things change behavior. Generally what we found was that the pesticides at high concentration was sort of making the fish sluggish. They generally traveled shorter distances. After you simulated a predator attack, they didn't try to get away as fast as the fish that weren't exposed to pesticides. And they had lower mean and maximum swimming speeds after the simulated predator attack. But I'm not even going to bother showing you the graphs of what the parasite did, because there was nothing. No interaction with the pesticides, no nothing, and I have repeated this experiment 3 different times, focusing just on the parasites, and there seems to be no effect of the parasite on those behaviors that I talked about. So it suggests that parasite manipulation is extremely specific. And maybe you're thinking, Kelly, I can't fathom why you're surprised by that, because you told us at the beginning that those ants were doing that thing on the northwest side of the plant at solar noon. This shouldn't be surprising. To you, I'd say nobody likes it when people rub things in. Maybe I shouldn't have been so surprised. But it does seem that this is extremely specific, and it could be extremely specific because there are lots of ways that the fish can die that also kill the parasite. So if the fish gets eaten by another predatory fish, the parasite dies, and so does the fish. If the fish can't compete for resources with conspecifics, both the fish and the parasite die. So there might be strong selection pressure for only particular behaviors to get changed and to just generally let the fish go about the job of staying alive on its own. So anyway, looks like manipulation is extremely specific. That was disappointing to find for my PhD, but whatever. Okay, so for those personality traits, turns out that the parasite doesn't seem to be changing them much, but there are still those conspicuous behaviors that are interesting, and additionally, in 2012, Friedensborg and Longoria were looking at a very similar system in the Gulf of Mexico. So it's the long-nosed killifish, which is a very closely related killifish to what I study. And there's a Euhaplorchus species, which is a trematode in the same genus as the trematode that I study. And in that system, infection by the parasite causes the killifish to shoot to the water surface very often, like we see in our system. That's one of the conspicuous behaviors. Additionally, infected fish just sort of generally hang out near the top of the water column more often than the uninfected fish. So you can imagine that maybe that benefits the parasites, because if the fish are higher up, they're easier to catch by predatory birds. No one has tested that. That's a story right now. But we became interested in looking at how microhabitat choice, specifically where the killifish decide to hang out in the water column, how that's affected by the parasite, and whether or not there might be feedbacks. The reason we thought there might be feedbacks is because it looks like the parasites swim up to the top of the water and aggregate there, and then the killifish that are infected spend more time hanging out at the top of the water column, where they acquire more parasites, and then spend more time high up in the water column, blah, 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 you see where I'm going. So as a behavioral ecologist, I'm interested in whether or not you can predict the means, the max, and the distributions of behaviors in a population. So let's consider what worlds look like hypothetically in worlds where you don't have these feedbacks and worlds where you do. So along the x-axis, we have two different scenarios. Does behavior influence infection risk? If the answer is no, then the killifish, for example, the killifish encounter parasites sort of randomly. Anyone is equally likely to encounter a parasite. If the answer is yes, then individuals who exhibit more of this behavior have a slightly higher risk of getting infected, slightly higher risk of encountering parasites. Over here, on the y-axis, you have parasites influence behavior. If the answer is yes, then the parasites shift the behavior of the host. And if the answer is no, then the parasites don't change behavior at all. Okay, so up in this first quadrant, you have a situation where behavior doesn't change, but over time, and time is shown by the circles becoming more dark green, over time they acquire parasites. Acquiring parasites doesn't change the behavior of the host. Everyone just has more parasites over time because they've lived longer and acquired parasites kind of randomly. In the situation where behavior influences infection risk, individuals who are sort of over more to the right here exhibit more of the behaviors that get them infected, and you end up with a positive correlation between the number of parasites and the behavioral trait. In a world where behavior doesn't influence infection risk, but parasites do influence behavior, you again get sort of a correlation, a positive correlation, but you also start getting shifts in behavior over time. So in this population, depending on when you take a snapshot of the behavior across ontogeny, you get different means in the behavior over time. But over here, this is what positive feedbacks look like. Behavior influences risk of infection, and the parasites are causing more of that behavior over time. And with these feedbacks, you end up having, one, a lot more variability in the behavior, a different mean of the behavior, and you also have different maximum levels of the behavior. And you also have a lot more parasites in the system, because a lot more of them are surviving and getting picked up by the host instead of dying in the environment waiting for them. So you can imagine that these sorts of feedbacks could have important implications for population dynamics for both the host and the parasite. So we currently have a grant that, who knows how successful grants ever are these days, to do an experiment that will really nail this stuff down. But we have some indirect evidence that we live in this world. And so I'm going to talk about some data we have already and some preliminary data that we have that I'm excited about. So first, one of the first things you'd want to know is where are the parasites? Where are the parasites hanging out? Are they actually in the top of the water, or are they hanging out somewhere else? You'd want to know if infection increases the time the hosts spend in those high-risk areas. You'd want to know if infected individuals are more likely to acquire additional parasites. And you might want to know, why does this even happen? So like I mentioned, the parasites can get up to intensities of 8,000. Like, shouldn't you have crazy competition happening at that point? Like, literally, the parasites, there can be so many parasites that they make up 0.5% to 1.7% of the biomass of the fish. That would be like a 150-pound person having 3/4 to like, what, 1.5 pounds of parasites on their brain. That's pretty creepy. You'd have to imagine those parasites were starting to compete at that point. So okay, first we wanted to know where the parasites are. So these parasites, during their free-living stage, are pretty tiny, and they have no chance of catching up with the host. The hosts move much quicker, so the best that they can do is end up in microhabitats where they're likely to encounter killifish. So we tried to figure out what cues are they using to determine where they end up in the water. So we made this super simple apparatus with 3 compartments where you could shine the light on it from below or from above, and we just looked to see where the parasites go. And then you can also look at how they respond to gravity by just seeing what they do when you shut the lights off. And what we found was that when you turn the lights on from above or when the lights are off, the fish move up into the top and the middle compartments. So you find more parasites moving up when the lights are on from above or they're off. But when you turn the lights on from below, the fish go down to the bottom compartment. So they are moving towards light. They move away from gravity in the absence of light, but light is the cue that they really use to decide where to go. So presumably they're aggregating in the top of the water column in nature as they swim towards light. We realized that much better data would be to figure out where the parasites are in the estuaries by sampling the water. So we had some engineering students build an apparatus that was supposed to do that, but after an entire summer of trying to make it work, we had to call it quits and do this for the meantime. So hopefully we'll get those data where we actually sample the estuaries. But in the meantime, we have this indirect evidence that parasites are likely aggregating near the water surface by moving up towards light. So then we wanted to know if infection increases the time that hosts spend in high-risk areas. So we already know that infection makes the fish shoot to the surface of the water. That's one of the conspicuous behaviors that's been quantified. And we know in that other system that I talked about that fish just sort of generally hang out near the water surface. And so we wanted to make sure that was happening in this system as well. So we collected juvenile killifish from a population where you don't find the parasite. And so they're uninfected by the parasite. And what we did was we measured their behavior before they were infected. And then we infected half of them and waited 42 days to measure their behavior again. 42 days is more than enough time for the parasites to become mature enough to transmit to the next host in the life cycle, and enough time for them to reach their asymptotic growth rate. So at that point, we should expect manipulation to be happening. This is another one of those experiments where I wanted to measure a ton of different personality traits. So I looked at activity, boldness, and social behavior, and controlled for order of assays and time of day. And none of the rest of that data is worth talking about, because we didn't find anything. But the one thing that we did find is that if you look at the difference in the amount of time that fish spend in the top of the tank, it increases for fish that were infected by the parasite. So over here, you see that the second time that you measure the behavior of our control fish that didn't get infected, they don't really change the amount of time that they spend in the top of the tank. Maybe they decrease it a little. Whereas when you infect the fish, the second time you measure their behavior, this is only over a 3-minute period, they're spending a little bit more time at the top third of the tank. So when you extrapolate out over the course of the entire day, that could translate into a lot more risk of infection. And additionally, I'm not going to show any data from this experiment today, but we have hatched the killifish from eggs in the lab, and every 2 weeks we've been infecting them with parasites like would happen in nature, although it would actually be every day in nature, but we have time constraints. But anyway, this effect becomes even more pronounced as the fish get older and get more parasites. So this is a robust, replicable finding.
I just have a question.
Yeah.
Are the fish responding to the same environmental cue? Are they using light? Is this aquarium like dark around the sides and light on the top? Or
So it was the parasites that were responding to light.
Sure,
but
right. But the fish, in this case, are they responding to the same light to get to the top, or is there like a temperature gradient that fish?
the tanks were the same
Were the same, I
both
guess.
times,
Yeah.
and we had temperature as a covariate in the model to try to control for that stuff. We did, they were in the exact same tank that was in the exact same part of the room, so hopefully everything was exactly the same in between measurements. So it does look like they just sort of generally are moving up, and I don't know if that's in response. I don't think that's in response to light, because they were in a greenhouse where the light was coming in from the front of the rooms, and I don't think we had the overhead lights on. Okay, so it looks like the parasites are aggregating at the top of the water. Looks like when fish become infected, they spend more time in that area. So then we wanted to know, are infected individuals more likely to acquire additional parasites? So as part of that same experiment I just told you about, we took 5 groups of 24 fish, and the 24 fish had been living together. Half of those fish were infected as part of the experiment, and half were uninfected. And we put them in these mesocosms. And these mesocosms have a flywheel that generates current like what you would see out in the estuaries. And we put snails that were infected by the parasite in these mesocosms so that the snails could naturally release the parasites over the course of 3 days. So over 3 days, we just kind of let the fish move wherever they wanted. And one of the things I love about this system is that it's possible through looking at morphology to figure out about how old the parasites are. So right when the parasites infect the host, they look like this. They have photoreceptors, these little black eye spots, and yet again, I study really cute parasites. And so these guys have cute little eye spots, and they're shaped kind of like eggs, and there's 4 of them here, and they're pretty small at this stage. After about a week or 2, they look more like this. They have a more defined cyst. They're longer, so they're more curled up on themselves. They don't have the photoreceptors anymore. And by the time you start doing the dissections, this is different lighting, doesn't show up so great here. But here's what the adults look like. They're ovals with clear cysts. They have a white spot that's the excretory bladder. And there's one of the new little infections up there. Can you see there's 2 little sets of eye spots? Right in between there. Anyway, take my word for it, they look different. And so, okay, so these are the 2 treatments. So these are fish that were not infected before the experiment or as part of the experiment. These are fish that were infected as part of the experiment. This is the number of new parasites they picked up while they were in the mesocosms. Contrary to our predictions, the control fish acquired more parasites. So this could be a story about the adaptive immune response. These fish had 42 days to mount an adaptive immune response, so their immune systems were ready to go the next time they encountered that parasite. It could also be that after one encounter with the parasite, they were a little bit better at avoiding the parasite in the future. However, this doesn't really tell you much about what's happening in nature, because as you might remember, these uninfected juvenile to adult sized fish just don't exist in nature because they're already infected. So the more relevant comparison here is what happens between fish that differ in the number of parasites they had going into the mesocosms. This is a super preliminary look at the data. So on week 5, the number of competent UHAs, so this is the number of parasites that are ready to infect the next host in the life cycle, they're over 45 days old. The number of parasites we achieved in those infections as part of the experiment were a bit higher than we achieved in weeks 1 through 4, so I put them in a different graph just to make it easier to see. But here we have a positive relationship between the number of parasites that a fish had going into the mesocosms and the number of new parasites that they picked up when they were in the mesocosms for those 3 days. And for weeks 1 through 4, we have a positive correlation in 3 of the weeks. We have a negative correlation for this 1 week where we only acquired really low infections as part of the major behavior experiment. So we have some evidence here that there might be positive feedbacks, and we have an experiment that I wrote the grant on where we're hoping to measure exactly where the fish are in the mesocosms over time and try to get a much better handle on this. But so at the moment we have evidence that the parasites hang out on the top of the water, that infected fish spend more time there, that fish with more heavy infections are picking up more parasites when you put them in these semi-natural environments. And so then you might want to know, what's happening with the parasites when you get to these really high infection intensities? As I mentioned, there can be about 8,000 parasites on the brains of the fish. Shouldn't that be bad for the parasites? Shouldn't they have some competition at some point? And so what we did was we collected wild-caught fish that were naturally infected. We measured the volume of the parasites, and we looked to see how the volume of the parasites changed as the number of parasites increased on the brain. So this is the density of parasites, so the number of parasites per gram of fish. So these fish were like 2 to 4 grams, and fish over here had about 1,600 parasites on their brain per gram of fish mass. And the open circles are the volume of the parasites within a particular fish. The black circles and the black line are our model average predictions. And as you can see, there's no negative relationship. So there's no sign that even at these really high densities, that the parasites are competing, despite making up 1.7% of the biomass of these fish. So it looks like they have more than enough resources to do the growing that they need to do. And additionally, there might be a slight positive relationship where parasites that are found in higher density infections are actually bigger. And the size at this life stage is important for knowing how many eggs these guys are going to produce when they become adults. So in other systems, they found a really nice correlation between the volume that the parasites achieve in their hosts and the number of eggs that they produce when they get to the next host in the life cycle. Does that make sense? So the fitness implications of competition here can be really important for their lifetime reproduction. So this 95% confidence interval just includes 0. So it's possible that there's no benefit, but at least they're not suffering from competition. But if there is a benefit, what might that benefit be? So we suspect, we being the field of manipulation, we suspect that manipulating the host is costly. So if they're producing, for example, neuroactive compounds to change brain chemistry to induce these changes, that might be energetically expensive. And you can induce more manipulation at less cost when you're in these high density aggregations. So maybe they're sharing the cost of manipulating at these high densities. Alternatively, we know that a lot of helminth parasites can suppress the immune system of their host to increase the survival of the parasite. So maybe it has nothing to do with manipulation and everything to do with suppressing the immune system so that they can all survive. But again, they can cost share there and all invest more in their own growth and do a little bit better in high groups.
Just to clarify, are these all the same type of parasites?
Mm-hmm.
Okay.
Yeah, and so there are other trematode parasites infecting killifish, and we looked at one of the parasites that lives in their liver, and you don't see the same relationship. I think they also did not show evidence of competition, but they didn't have a strong or a positive relationship like this. So it's not necessarily happening with all of the okay, so parasites seem to be aggregating near the water surface. Infected hosts spend more time in that high-risky area. And it also seems to benefit, and these potential feedbacks seem to benefit the parasites, both because they might be reaching slightly higher volumes, but for also the reasons that we talked about earlier, where higher density infections induce more of those conspicuous behaviors and make it more likely that the host gets eaten by predatory birds. So in these high density infections, the fish are more likely to get transmitted to the next host and have a chance at reproducing at all, and they might even be bigger when they get there because they benefit from cost sharing inside of the host. So if these positive feedbacks are occurring, the parasites are likely benefiting from them.
So actually, 2 quick questions. The what is it? Suppressing the immune system theory, wouldn't that also create an environment for freeloaders? So like if you're not actively fighting the immune system, but everybody else around you is, shouldn't you grow larger or something along those lines?
Yeah. Sorry, go ahead.
And then, sorry, the next one was with regards to the last graph that you showed. Is there a correction for the size of the fish itself? So do you just get bigger parasites in a bigger fish?
Do you just get bigger? So standard length was a covariate in that analysis. You don't just get bigger parasites in bigger fish. And then for your free rider question, yeah, so there definitely could be hitchhikers in this system, and additionally there are other trematodes that get transmitted to the predatory birds when the fish get eaten. We don't think that they manipulate, and we do think of them as hitchhikers on Euoplochus californiensis, which is doing all the hard work. And because this parasite is so ubiquitous and all the fish are infected, the parasites, for example, the ones that live in the liver, can be sure that they're going to end up in a manipulated host because the parasites are everywhere, and so they just kind of hang out and wait for it to happen, we think.
Sort of related question along those lines. Have you or others looked at the genetic diversity of the parasites?
No, good question. So no, we haven't. So some work has been done trying to figure out if the fish are getting all of their parasites from like one snail, or if they're getting them from a lot of different snails. So
they are sexual, right? The parasites? Did you say?
They're sexual when they are in the birds. They're asexual in the snails. So if a fish did hang out just like on top of the same snail all day long, it could have all of the exact same genotype. But these fish, so like if you stepped your foot in the salt marsh, a foot about my size would probably be squishing about 25 snails. These snails are everywhere, and a lot of them are infected by the parasites, and these fish go up and down the channels all day long. So nobody has looked at the variability. It's possible that there's a lot of the same genotype out there. They probably are still acquiring parasites from lots of different infected snails, but nobody has gone out and specifically, as far as I know, looked at the genetic diversity. So I mean, it could be, if the genetic diversity was pretty low, then it could be that they're cooperating because they're surrounded by kin. My gut tells me that's not what's happening, but I don't have any data to back up my gut. Yeah, sure.
It's more of a comment than a question.
Yeah.
Cliche. But yeah, regarding the free rider comment, because they're not reproducing in the fish, it seems like you couldn't really have selection for free riding versus cooperation because you have no ability to have any prediction or respond to the immediate environment of the brain. You don't know what kind of brain you're going to end up in and what the concentration of free riders versus cooperators would be if there are phenotypic differences.
So there was a postdoc who never published these data, but there's some evidence that the parasites secrete something so that they can find each other when they get onto the killiefish's brain. And so I agree that there's no way for the fish to know, the parasites to know what kind of fish brain they're going to get on. And I think almost all of the parasites die in the water waiting to find hosts. But once they are on a fish's brain, if those chemicals are being released, they might have some way to figure out like about how high of a density they're in, and they could make decisions based on that.
Right, so it'd have to be but there'd have to be some sort of behavioral plasticity rather than just like a phenotypic default behavior. Okay.
Yeah, there could be plasticity. Okay, so basically what I'm saying is we think that we live in this world where the parasites are influencing the mean, the max, and the variability in behavior over time. And these sorts of feedbacks might explain why this parasite is one of the most abundant parasites in systems, despite infecting only one of the hosts in the system. And it might explain why some of the fish can have 8,000 parasites, which is way more than any of the other trematode parasites that they're usually infected with by number. So we think that we exist in that world. And I was going to stop here, but there weren't as many questions as I thought, so I still, go for it.
Can I ask one question?
Yeah, please.
Does the killifish, is that an instinct that we know of in the absence of a parasite? So for instance, to go to the surface and flip to the side. My comment comes from the toxoplasma literature. So like, the parasite really in the end is accessing an already innate programmed fear response. For toxo, it's shutting it off, but I'm wondering if what we're seeing as manipulation is already there, instinctually encoded somewhere, but we see it just more often with the infection.
So first, a comment about the toxoplasmosis system. I think we believe currently that it's more than just shutting it off. It's like reversing. So instead of being repelled by the smell, they're actually attracted to it.
They are attracted.
But still, it's along a gradient of pre-existing. But so in this system, yes, uninfected fish in systems without the parasite do these behaviors, but they do them far less frequently. So the parasites in this case do not seem to be creating new behaviors. They seem to be co-opting pre-existing ones. So for example, fish will often rub against hard substrates to get off ectoparasites. That's one of the conspicuous behaviors that we see. Shooting to the surface of the water happens sometimes in uninfected fish, but it's not something they would usually do unless there was food up there, whereas the infected fish do that pretty often. The fish also do this like weird contortion where they turn in the shape of a C. That's a common thing to do before they try to escape a predator, so they very quickly go in the shape of a C and then use that to like shoot forward to try to get away. They also do this weird twitching behavior, which I have not yet figured out like why uninfected fish do that. And then there's another behavior they do that's part of their mating season that happens completely outside of the mating context. So yeah, these are all pre-existing behaviors that are sort of induced to happen more often. Yeah. More questions?
So one of the things that draws our attention to these cases is how big the cycle is.
Mm-hmm.
And you would think that you could have a parasite that once it escapes the snail just goes and buries into bird poop, and the cycle's much smaller, and you wonder, why is this cycle so big? And one hint comes when you said that sexual selection or sexual reproduction only happens in the bird.
Mm-hmm.
Why is that?
So we don't know for sure the answer to that. So we have some conjectures. So for example, we know that in a lot of other trematode systems, fish will actually eat these parasites, the free-living stage. And in some cases, that's important for the transmission of the parasite. But if the parasites, for example, started their lives in the snail and that was their main host, then they went off and had a free-living stage to go in search of more snails to complete their life cycle. You can imagine that there would be a selective benefit for the fish, or for the parasites, that could survive getting eaten by the fish. And if there were enough fish that were eating, these are essentially just little packets of tons of energy, so they're probably pretty nutritious. So if there were enough predators that were causing enough mortality by just eating them, maybe there was a selective pressure to be able to survive inside of the fish, and that's how they captured another host in the life cycle. And then they got eaten by a lot of birds, and so they moved into the birds. And actually a great thing about those birds is that they fly from estuary to estuary, spreading your eggs far and wide, which is something that would be great for the parasites. And anyway, so that's the current going story for how this complicated life cycle arose. But I don't think we know for sure what happened. But it's fun to think about. Any more questions? Okay, then I'm going to keep talking, unless everybody wants to go, which is fine also. But it's 4:30. Should I keep talking or should I let people go?
I vote keep talking.
I'm going to keep talking.
Encore.
Okay, encore. Here we go. All right, so next.
From the top.
Thanks for the suggestion. Okay, so I'm also interested in the mechanisms through which parasites manipulate host behavior. A lot of this work that I've been doing has been done with collaborators. So we recently got a grant from the Norwegian government. They really love funding fish stuff, apparently a lot more than my government does. And so we've been getting a lot of our money for this stuff from them. And so through that project, we're doing genomics, metabolomics, proteomics. We're focusing in on particular aspects of the immune system and particular measures of metabolism. But unfortunately, you don't get to hear about any of that today because we just started that. What I will tell you about is what we've learned about the hormone cortisol, which is a stress hormone. So I will remind you that Jenny Shaw did some work finding changes in serotonergic and dopaminergic activity in fish that were infected. Relative to uninfected fish. So this is just a cross-section of a brain of an infected fish. All of these little black dots are the parasites sort of sitting on top of the brain. They're not inside of the brain tissue, but they're sitting on top of the brain. And then on that brain, you see none of the black spots, so that's what an uninfected fish brain looks like. Mostly I just show that because it's awesome. So we know that serotonergic activity is changed in response to infection. And in fish, serotonergic activity interacts with the hypothalamic-Pituitary-Adrenal Axis, which is analogous to the hypothalamic-Pituitary-Adrenal Axis in people. And essentially what you need to know is that this axis is associated with the release of a stress hormone called cortisol. And if you are getting chased by a lion across the savannah, you release cortisol. Cortisol helps you liberate energy so that you can run as fast as you possibly can. It shuts down digestion because you don't need to worry about that steak right now. You just need to worry about survival. And in teleost fish, it's also associated with a bunch of different locomotory behaviors. And so we thought, well, maybe cortisol is important for inducing shooting to the surface behavior, or that scratching behavior that we talked about. Usually they shoot forward really quick before they do the scratch. So maybe cortisol is associated with some of these conspicuous behaviors, and we would predict that it should be changing based on what we know about changes in serotonergic activity. So based on what we know, we predicted that UHOS should suppress baseline and stress-associated cortisol. So to sort of start to get a handle on this, we collected wild-caught adults that were already infected by the parasite, and we took advantage of this really awesome feature of fish, which is that hormones like cortisol and their sex steroids leak across the gills and into the surrounding water. So if you wanted to draw blood from this fish 4 times, you definitely kill it. But you can stick it in a beaker of water 4 times, collect hormones, wait an hour for the hormones to reach equilibrium, pull them out, and then measure the hormones in the water, and that's strongly correlated to the hormones that are in the blood. So a nice non-invasive way of measuring these hormones. But first you need to validate the technique. So we collected killifish from the wild, we marked them with visible implant elastomer tags so we could track them through an experiment. We split them into 3 tanks, and every day for 4 days we measured their hormones. While we were doing that, we also measured what I call handling time. So this is the number of seconds it took for me to catch a fish with a net, lift it in the air, and then stick it in a beaker. So handling time is a within-day measure of how stressful the procedure is for the fish. And it turns out that on the different days of the experiment, fish weren't consistent in how long it took to catch them. So there weren't some fish that I always caught really quickly and some that always took a long time. It's the handling times were sort of evenly distributed for the fish. Okay, so we did that on 4 days. We measured the hormones using enzyme immunoassays, and then we found a significant interaction between the density of parasites and the amount of time it took for me to handle the fish. And nothing else was significant, so no baseline effects of the parasite, but an interaction. So what we did was we plotted our model predictions for 3 different densities of parasites. So this is the first third of a triptych plot looking at low UHA density fish. Down here we have standardized handling, so numbers over here, it took me longer to catch a fish, and here it took me shorter. This line represents the average behavior of fish with low UHA densities, and over here is the log cortisol release. So we were a little surprised to see that when you do a lot of handling, low UHA density fish produce less cortisol. So that's contrary to what you would usually think about the relationship between cortisol and stress, but it actually fit with the predictions I just said a couple slides ago, where we thought that changes in serotonergic activity might be suppressing cortisol response. So in my head, I was like, oh, this is so cool. The parasites are getting the fish to not stress out like they should, and maybe that causes them to respond inappropriately when a predator is around. This is the first time I've been right during my PhD. This is awesome. The next plot at medium densities, we're going to see an even steeper negative slope, showing that this effect is even more pronounced. Of course, that's not what happened. So here it looks like it's kind of evening out, and at high densities of the parasite, you start getting the positive relationship that you would usually expect to see between handling stress and the density of parasites. Cortisol is a notoriously difficult hormone to link with behavior in teleost fish. Sometimes it does what you think it will, sometimes it won't. But interestingly, we just looked at a bunch of uninfected fish, and this is how uninfected killifish respond to handling. They show this negative relationship, and then it looks like at higher infection intensities, you start inducing this positive relationship. So now I'm going to tell you a new story that fits the data, which is that, hey, maybe when they're getting chased by a predatory bird, they release more cortisol, and that causes them to do more surfacing behaviors, blah, blah, blah. We're working on making hormone implants so that we can manipulate cortisol levels and measure changes in conspicuous behaviors. But so as I told you earlier, you can easily convince yourself that changes that you see that are correlated to a parasite are caused by the parasite when maybe they're actually caused by the host. So one thing we wanted to do was convince ourselves that this difference in response to handling stress as parasite density changes is a function of the parasite, as opposed to just the way the fish responds to the stress of handling when it's also heavily infected. So to get a handle on that, we measured the cortisol response to handling stress at these different time points. So day 0 was we had a bunch of uninfected fish that we had collected from the field from a population without the parasite. We measured pre-infection what their hormone release rates were. Then we also measured at 7 and 14 days. And this is important because at 14 days, the parasite is able to infect predatory birds. But before that, it can't. So you shouldn't expect it to start manipulating its host until it would benefit from doing that. Because if it manipulates its host in this first 14 days, that's suicide. So our hypothesis was that if we start to see that change in hormone release rates in response to handling stress during this 14-day time window, then that's probably induced by the host. If we see it at 42 days or any time after the 14-day time window, that's probably a phenotypic response that's induced by the parasite. And in particular, at 42 days, the parasites reach their full size. So they reach the full size, and therefore, as we've talked about, they want to get as big as possible so they can produce as many eggs as possible in the definitive host, in the birds. So we wanted to know at 42 days, what do they do? So the interaction between handling time and parasite density becomes significant at 42 days, but not before, suggesting that cortisol is something that's changed by the parasite and is part of the story of manipulation. And so it's something that we're going to look into a little bit more. So this is what our control fish looked like. This is what happened with the fish that had higher infection intensities. So it looks like these cortisol changes are induced by the parasite, but now we're going to try to get a little bit more data to get a better handle on what the implications are for killifish behavior. So with that, I actually am done. So hopefully I've convinced you that parasites are creepy but still awesome. That when someone tells you a story about manipulation, you should listen to see if they actually have data showing that behavioral change benefits the parasite in some way, even better if they have mechanisms data. And hopefully I've convinced you that maybe we have some interesting positive feedback things happening in the system, which could be important for understanding the distribution of behaviors in the system, and important for understanding how fish can end up with like 8,000 parasites on their brain. So thank you very much for your time.
I have a slightly random question. And as a person who studies the human brain, local influence of the gut microbiome on cognition, right? So nobody knows why. I mean, you transplant bacteria from the gut of autistic mice and you put them in healthy mice and they start showing these bizarre symptoms of the autistic mice. So I'm just wondering, is all of this just an illusion? Like, I mean, maybe our behaviors are manipulated by things living in our gut.
So I have long been wanting to find a philosophy major who studies free will so that we can just talk all night about this. So the way I've come to like thinking about it is that your behaviors are a combination of what you want to do and the slight influence of all the stuff that's living inside of you. So we know that it seems that the gut microbes secretes neurotransmitters like our own that interact with our enteric nervous system. I think I'm getting those details right. I'm not a microbiota person. And so that might influence our behavior a little bit. Humans can get infected by Toxoplasma gondii. That correlates with changes in our personality. But that could just be differences in the personality traits between the kind of people who get infected and those that don't. I'm going on a tangent. I can talk more about that if you want. But anyway, the point is, it's nice to think that we're not alone. We're like a community of organisms, and our behaviors.
It's the royal we.
Yes, exactly, the royal we. I want to believe I'm mostly in control, and I think that's probably true. Yeah. But maybe to some extent, our behaviors are subtly changed by our little friends who are living with us, and hopefully our interests align most of the time. But that's not always true.
Right, now I'm also wondering if in the case of pathologies, that there's actually something more we could learn there, because
in the system?
In terms of neurotransmitters going out of balance all of a sudden, I mean if it's possibly has some kind of enteric origins, right?
Yep.
So anyway, there's lots of cool things there. Thank you.
Oh yeah, no problem. But I really like the sound of my own voice, so I'm gonna follow up a little bit. So there's a closely related small fish that is infected by a trematode that instead of sitting on top of the brain like mine does, actually lives in the brain, and it looks like the behaviors in that fish change when the parasite is at the stage where it's eating host tissue, and then it surrounds itself with a cyst, and then the host behaviors slowly go back to normal. And I've always thought that would be a really cool system to study how the brain responds to trauma and recovers normal behaviors after a traumatic event. And that's not even almost what you were asking, but whatever, it's interesting. Yeah, there you go. Yeah.
I have a related question that I like that you set up this sort of misassociation of mechanisms in the mosquito question, because there it does seem like, oh, we get excited about manipulation because it's cool, when really it's pretty much potentially just straight selection. Your question made me think of this, we thought maybe the malaria was driving the timing of mosquito behavior, but really the timing of mosquito behavior is related to infection, and the parasite is under selection there to be on time. Like, and so I wondered how you can tease apart those two aspects in your fish also, because it's tricky, because is reproductive timing of the parasite driving how it's manipulating the fish to behave? Or is the time scale of fish behavior driving selection on reproductive timing in the parasite?
Yeah. So that's a tough one.
Yeah, I mean, it's hard. Yeah, that's hard.
Yeah. So I mean, we've tried to figure out, like, to generally get a sense of the timing of the onset of manipulation by looking at it, like, at multiple time points after infection. But that's a good reasonable approach. I don't want you to not think that the experiment I just told you about is worth listening to at all. But in nature, those fish are acquiring parasites over their entire life. So when a parasite ends up on the brain of a fish, it's new, but it's surrounded by conspecifics who may have been there for long enough that they're ready to transmit. So there's probably selective pressure for them to, like, get ready to go as soon as possible.
Yeah.
Which might be part of why they're able to be transmitted at 2 weeks, but they don't stop growing until 42 days. So, like, they try to get the important thing out of the way and then finish the growth that they need to do. So that, these fish breed, like, once a year during a pretty small time window that's about about like a year after the time when they're born. Approximately. And so they have a lot of lifespan where they're just trying to survive to get to breeding. And they're the most abundant fish in these systems, so enough of them are surviving through breeding that everything works out. But yeah, getting a handle on the timing is a tough question if you want to do it in an ecologically relevant way, accounting for the fact that they always have a lot of parasites. And yeah.
I think there's many cat enthusiasts at CIS. I wanted to go back to your point about potentially different personality types being drawn to situations that expose them to toxo.
Sure, so I'll do the quick background on Toxoplasma gondii. So this is a protozoan parasite, and it reproduces sexually in the guts of cats. And then when it produces oocysts, and that's the stage that goes out in the cat's feces, and then can end up in your garden, for example, or anywhere where outdoor cats defecate. And then eventually the feces dry, but the parasites stay infective. So it kind of just ends up looking like soil. And if you garden and don't wash your hands well enough, or eat a vegetable that was in that garden and don't wash it well enough, then you can become the next host. From the parasite's perspective, it would be better if the next host were a mouse or a rat or something like that. But if it ends up in a human, it moves into the brain tissue, and it forms a stage that will essentially go dormant when the immune system finally shuts down its replication, and it will live in people for the rest of their lives. And if you give people personality tests, and then you look for correlations between the responses to those personality tests and their infection status, men who are infected tend to be more jealous and less rule-following than their uninfected counterparts. They also have higher testosterone relative to their uninfected counterparts. Women are more caring and soft. Like, essentially, they fit the stereotypes, for both men and women, they fit the stereotypes that you always hear about. And women who are infected have lower testosterone relative to uninfected women. And then additionally, infected people have slower reaction times, not to the point where you look at someone and be like, you're really clumsy, I bet you're infected. But if they're about to get in a car accident and they need to make a split-second decision, that really seems to matter, because infected individuals are 2.5 times more likely to get into a car accident than are uninfected individuals. Kevin Lafferty did a study looking at population-level average scores on personality tests and correlating it with population prevalence of the parasite. We have pretty good data on this, because if women get infected when they're pregnant, the parasite can go into the fetus and really mess up brain development. And so a lot of women, when they're pregnant, get blood tests. So at the country level, we at least know about what % of women are infected. And in Western countries, there's a pretty high correlation between prevalence of infection and how neurotic people in that population are, and a pretty good correlation with how much that population cares about meeting gender roles. But while that's all fascinating and really fun to talk about, it could just be that populations that really care about gender roles are also more likely to garden. And another way that people can get it is if it's living in the meat of, so like cows can also get infected, and it can live in just their muscle tissue. And if you undercook a steak, you can get infected that way if the cow was infected. So like, I think France has pretty high prevalence, maybe something like 60%, and I think part of that used to be because they used to like undercooked meats, but now I think that there's better food safety that weeds these things out before it becomes a problem, so you can still enjoy your rare steak. But so anyway, there are these correlations that suggest that the parasite is manipulating human behavior, but you'll never be able to do the experiments that you would want to do to nail that down, because ethics prohibits you from infecting people with this parasite and looking at changes in their behavior. So the best we'll have is correlations, which are interesting for stories, but don't tell you much. But the rats are attracted to the smell of cat urine, which presumably gets them eaten by cats. But that's another system where we haven't done the experiment to actually show that that gets them eaten by cats. But it seems likely that it does.
So with regards to that sort of question, I'm curious about effects that this parasite might have on the reproductive success of any of its hosts. So it seems like it's bad for the snail, but is it good for the fish?
Bad for the snail. We don't know if it's good for the fish or not. If on average it kills the fish after it has a chance to reproduce, and if, I don't know, maybe it induces that breeding behavior that's one of the conspicuous behaviors more often, maybe that makes the males more attractive, I really don't know. But for that parasite we were just talking about, rats who are infected by it are considered more attractive to females, and they're more likely to mate with infected rats, despite the fact that that parasite is also transmitted between individuals in the ejaculate. So the female can get infected by mating with infected males, yet she's more attracted to infected males than uninfected males.
Because it seems like, you know, these organisms do have to, or those genes that allow for these organisms that do host these parasites seem to have to stay in the system, or at least the parasite seems to benefit from their prevalence in their host systems.
Yes, but so that seems like a system ripe for cheating. So like if some UHA genotypes hold off on killing their host until the host has had a chance to reproduce, but others kill their host immediately, then that genotype is going to be able to finish a lot more cycles of its life cycle more quickly and get into the environment more fully than a genotype that like kind of waited it out. But if on average it just takes that long for infections to get high enough before they end up in the predatory bird, and just it happens that you can't get them in there before, you can't get the fish eaten before the breeding season, then yes, maybe manipulation of the mating behavior, if you can get that genotype that you know you can live in well because you're living in it now, if you can get more of that killifish genotype into the next generation, there might be some selective benefit for that. But I think you're more likely to see that in systems where reproducing is part of the story for how the parasite gets transmitted. So there's a virus that sterilizes crickets, yet the crickets are more interested in mating when they're infected by it. And so there does seem to be some like manipulation of reproductive behaviors by parasites that are transmitted sexually, which makes me happy I'm not dating anymore, because it's kind of like a creepy thing to think about in terms of like bar dynamics and whatnot. But anyway. Any other?
Won't at some point the snails run out if they're all being castrated?
So the snails, it seems like what happens is that the snails, the bigger they are, so the older they are, the more likely they are to get infected. But by the average age at which they're infected, they've still had plenty of opportunities to reproduce. And so somehow there's still tons and tons of snails in this system, and I think that's what's happening. They have a couple chances to reproduce, and some of them manage to become nice old adults without getting infected, despite the fact that there's 21 trematodes out there trying to castrate them. It's a rough life. It's good to be a human. Okay. All right.
Okay. Thanks, Kelly.